All right, well, I think we're starting to get ready. So I wanted to say hello to everyone and welcome to our first Science Cafe of this semester and our first Science Cafe that's going to be an online experience. Before we get started, I had just kind of a few announcements about how it's going to work in this format. So thank you all for being here. Due to the coronavirus and worries, concerns about that, all the science cafes for this semester are going to be online. Currently, our plan is we're going to be broadcasting them on Zoom, which looks like everyone got the link to, as well as on YouTube Live. Due to some limitations with these digital formats, I think what's going to happen is everyone's going to be muted by default when you guys come in. If you guys have questions for the speakers throughout the talk, you can submit them to, there's an account in the chat room, a participant name that is titled questions. So you can submit any of those as a message to that account. And then towards the end, I'll read those out at the end of those so that the speakers can go through and answer any questions that you guys have. This is my first time hosting a virtual talk, so if anyone at any point has any suggestions or comments, please feel free to either submit them in the chat to the questions account, or you can send them to my email, which is ryerson1@illinois.edu. All right, with that, I think we'll go ahead and get started with a talk from our University of Illinois iGEM team. So go ahead and take it away.
All right, thanks. I'll just go ahead and share my screen. So, hi everyone. Thank you for coming here today and listening to us talk. We are UIUC's iGEM team for 2020, and our project this year is called Viralizer. My name is Sachin Jaju, and I'm a senior studying MCB.
I'm Yan. I'm a sophomore in bioengineering.
Hi, my name is Royal. I'm a sophomore studying biochem.
Hi, I'm Angela. I'm a junior studying integrative biology.
Our other two members were unfortunately unable to make today due to conflicts, but our whole team is a team made up of undergrads, and most of us are interested in going into research or academia. So iGEM stands for the International Genetically Engineered Machine Competition. And it's a competition that this is dedicated to the advancement of synthetic biology. It occurs annually with dozens of schools participating from countries all around the world, with more than 6,000 students taking part each year. So there are various different tracks that a project can take. And this year, since we didn't have access to a lab environment, we decided to do our project along the software track. So our project concerns the coronavirus. And obviously, we all know that it's the virus that is responsible for the COVID-19 disease. The virus is part of the coronavirus family, and the current strain that's infecting humans originated in bats. As of right now, there's over 25 million cases and 850 deaths worldwide, with more than 6 million cases and almost 200,000 deaths within the US alone. So the virus is highly mutative, and this allows reinfections to happen because the spike protein gets repeatedly mutated and permits the virus from infecting the same person more than once. And the structures for these mutated proteins are not solved, and there's not too much research that currently exists, not too much published research that currently exists that focuses on solving the mutated proteins structure.
Okay, so our project, the main focus of the project is on the spike protein. So on the right, you can see is a rendered model for the spike protein that we generated on our own. So the spike protein is one of the membrane receptor proteins on the coronavirus. It is the protein that is responsible for attacking and hijacking and entering human cells. So this is a very important protein to research on, whether you are trying to develop drugs or you're trying to develop vaccines towards it. So many labs, hospitals have published their sequence data from the patients that got infected for the coronavirus. And we can see that this surface protein is constantly mutating and there's over 1,000 mutants for this specific protein. So currently we have a lot of vaccines that are targeting this protein specifically, but there's no vaccines that are developed to target all of those mutated spike proteins. So they might not be effective to all of the different strains of the coronavirus. And that is a problem that we're trying to tackle.
So for motivation, early on, we noticed that all of our lives and college experience was greatly affected by this pandemic. And this was one of the motivations behind Viralizer. And currently, there's only 10 out of thousands of spike proteins that have their crystal structure solved and published. And we hope that with this project, we hope to help scientists in their research efforts outside the current lab. And currently, there is no database that focuses on spike protein mutations or shares mutated protein models. And there will be an upcoming slide that shares more information about this.
So we had a few concrete goals that we wanted to achieve with this project. The first was to create a database of spike protein sequences that anyone could access. That's just online. Another is that we wanted to characterize the propagation of the virus as it spread throughout the world. We also wanted to enable researchers to use our tools to visualize novel protein models, to analyze structural effects of possible mutations. This would allow them to see how those mutations could affect antibody binding and would help with the development of a neutralizing agent or vaccine. And another final goal of ours is to develop our own antibody or neutralizing agent and hopefully test it experimentally.
Okay, so this is just a very simple flowchart of our project. So all of our data that we're going to show later starts from the spike protein sequences that are available on the database online collected from real patients. And so we condense the sequence because there's just too much of them. We start with about 70,000 sequences, and we try to find sequences that has the mutations in the spike protein that we want. If it's in the database, we will just ignore it and just update like the case density for the specific strain. If it's not, and then we're just gonna start scraping those protein sequence and put it into our sequence database. We're also gonna see if that specific mutation is gonna change the structural stability of the protein. If the protein structure is not stable because of the mutation, then we will just computationally calculate a model for that protein using a method called loop modeling. And then we're just gonna put that model into our own database. And also, we're also gonna try to design our own antibody computationally. So because we have all those variants of the spike proteins, it will be very valuable to see if we can develop, at least on our computers, an antibody that can bind to all these variants of the virus. So we start with a protein file that contains both the antibody and the spike protein. And we using a bunch of methods, and we try to come up with several good antibody designs that researchers can test out into their web lab. And all these data will be visualized and stored inside our web page, and it will be open to the public and to the researchers.
So this is a visual of the three main aspects of our software. The first being database, and this stores mutation data from modeling point mutations. It will also store the top antibody designs. The second is modeling, and this allows the design for point mutations and associated heat map diagrams, which will be on a future slide. And finally, phylogenetics, which will comprise the phylogenetic trees that show the spread of COVID-19 throughout this pandemic. And it will also provide other data such as geolocation, strain, and other information. For the main website, we use HTML, CSS, Bootstrap, and cPanel, as well as some JavaScript.
So I'll cover the phylogenetics aspect first. We built a phylogenetic tree to display the variation in viral genome over space and time. Using an open source tool, NextStrain, which allows interactive visualization. The sequences are from GISAID database that contains worldwide genetic sequences of coronavirus and associated epidemiological and clinical data. Looking at a simple next strain on the right, the horizontal axis measures the divergence of varying strains based on sequence differences. More available on the next slide. So this is the page of tree and the model. On the right first, the current tree is built with the pre-selected 234 sequences from GISAID that had a point mutation that led to structural change in spike protein. These sequences appeared more than 8 times, and all sequences appeared in July. The user can color the sequences by country, sampling date, and genotype. The live map on the right top shows the transmission routes of the virus. On the bottom of the right, the user can view the specific protein mutation in sequence fragment using the diversity panel. And on the left, we used open source molecule visualizing tool called Molstar Viewer to embed our sequence and the corresponding protein model we generated into our website. So overall, the visualization allows us to view the bigger picture and the context of our data by characterizing the propagation of the virus. This is the live map based on our tree. And it's gonna show the map and the tree on the a tree together.
Okay, so sorry for that. Another very important part. Gosh, yeah, it's just motorcycles on Green Street, sorry. Another very important part of our project is the modeling part. So on the left, you can see a protein, which is color coded by, you can see the spike protein with the surface color coded by the number of mutations happen on that specific region. So red corresponds to less than 10 mutations, white corresponds to mutations, no mutations, green corresponds to less than 100 mutations, and blue corresponds to less than 1,000 mutations. So in total, we have about we have about several thousand different variants. So after we figured out where does the mutations happen, the next part is to think what actual protein models are we trying to generate, because calculating those protein models on a computer is a very time-consuming job. So on the bottom, you can see a heat map that represents what mutations can greatly affect the structural stability of the protein. So the a red box simply means that a specific amino acid mutation on a specific amino acid index is going to greatly affect the stability. And for these red boxes, we're just going to calculate those protein models on our computer so that we can save some computational power. So on the right is a direct comparison of our modeling results with a current state-of-the-art online protein modeling service, which is called SwissFold. And you can really see that we're comparing to a same loop on a specific on a part of a protein, and you can really see that our structure is much compacted compared to the online generated results from SwissFold. So for and also another very important part of the project is the antibody design part. So we are trying to design several versions of antibody on our computer that researchers can test on them. So for that, we first run a point mutation scan on the protein file that contains both the antibody and the spike protein. The way that point mutation scan is going to work is that we're going to mutate random sites on the antibody and see how that is going to affect the binding affinity of the antibody with the spike protein. So if an antibody can bind more firmly to the spike protein, that means that that specific mutation can probably increase the effectivity of the vaccine. So after that, we're just going to use a very advanced algorithms called Gaussian process regression. And then we're going to use our point mutation scan data and try to come up with several good designs for the antibody. And after that, we're going to use all of the spike protein variants we have and then iterate through them using our antibody designs and see which of our design can bind to as many variants of the spike protein as possible. So this is the final and this is the final goal of our project. And this is the part that we're tackling right now.
So some of the future goals of this project, as we said earlier, the first one would be to finish designing the antibody and test its efficacy in the lab. We also want to allow users of our tool to model their own sequences rather than just have the sequences of the spike protein that are currently in our database being modeled. We want to allow the users to be able to model their own sequences as well. We also want to expand and update the database as well as the phylogenetic tree with new sequences as time progresses and the pandemic progresses as well. The last thing that we want to do is talk to end users about our website, such as researchers and doctors, to see how we can adapt the database as well as the modeling and all the other components to suit their needs the best.
What we learned through this past summer and currently this current semester, we've learned a lot of open source tools as shown on the right. We've also expanded our current knowledge of viruses in general. And in addition, we've also interviewed many U of I faculty members who are on the front line researching COVID-19.
Lastly is acknowledgement that concludes our presentation. We just want to say thanks to our mentors who are a part of CRABI, as well as the external mentors who we reached out to for all their help with this project, because we wouldn't have gotten very far without a lot of help. So yeah, thank you. And thank you all for listening.
Cool. Thank you guys very much for your presentation. So far, we've had a few questions about asking more context about the iGEM team. Like, what is iGEM? Who can participate in iGEM? Like, what's the general timeline for projects on those? And how is your team assembled, the UIUC team?
Sure. So I can go ahead and answer some of that. So iGEM is a privately organized competition. They have different levels of teams. So there can be graduate teams, there can be undergraduate teams, and they can also be high school teams. And all of them kind of compete in their own separate sections. They're not all judged together. So yeah, we are part of the undergraduate section. And the timeline for the project is usually we start coming up with project ideas. In a regular year would usually be in April. I was actually part of the iGEM team last year as well. And we started coming up with a project idea last year, maybe in March or April. We do some work leading up to the summer on how to prepare ourselves for, in terms of researching the project, as well as the materials and whether it be feasible in a given time. Over the summer is when the team would do the bulk of the work, get done with the majority of the project. Usually this would be lab work, but since we didn't have access to a lab over the summer, we moved to computational projects so we could all work from our homes. And then after the summer, we mostly work on our wiki, which is separate from our website that we've discussed in this presentation. We work on our wiki, as well as finishing up any odds and ends that are left in the project in time for the Jamboree, which will be occurring mid-November, which is basically where all the iGEM teams from across the world come to participate and share their research, which will be virtual this year.
Cool. So another question that we had was, do you have any specific plans on how you're going to be reaching out to doctors to get input on your database? Are you going to be talking to people at Carl, or do you have contacts already made, or do you know who you're going to be talking to?
So one of the other members of our team has previously either worked with a physician or took a course under a physician at Carl. So she, Mary, will be able to reach out to the physician at Carl and hopefully get some input there. As for other researchers, we're planning to reach out to some of the people we've already reached out to, some of whom are all on this list. as well as we're also trying to reach out to the shield testing team on campus, though that's so far been unfruitful since they're obviously very busy, but we'd hopefully want to reach out to them too and see if they'd be able to use our project at all.
Cool. Are there any other questions that anyone has? Or anything else? Anyone, any comments or questions for the team? Well, if not, I guess I'll thank you guys again for presenting, and I'm excited to see how the work continues. And yeah.
Thanks for coming.
Thank you.
Thank you everyone for coming. We're going to continue on with these. So they'll be digital for the time being, and then we'll keep, if anyone has any suggestions on changes or anything that might help them, please let us know. All right. Thank you all.